This is the Cornellison Lab in Bond Life Sciences Center. They are part of the biology department, and the focus of their research is signaling and activity of skeletal muscle satellite cells. Satellite cells are adult muscle stem cells. When activated, they proliferate and differentiate before fusing to the muscle fiber for repair. The satellite cells, shown in green, are moving on muscle fibers shown in red. Satellite cells are harvested from the leg muscles of mice. Here the muscles are being separated from fat, bone, and other tissues. The muscle must be chopped to allow the target cells to be filtered from unwanted cells. A mixture is made containing enzymes to help break down collagen and connective tissues. The mixture is then incubated for an hour. Filtering the mixture separates the muscle cells from other unwanted tissues. A centrifuge is used to spin down the muscle cells, separating them from the liquid used during filtering. The cells are resuspended in nutrient-rich media and are plated onto a clean dish. This mixture contains the things necessary for the cells to continue dividing. The cells are then incubated overnight to mimic their natural environment. To look at the natural factors that might make stem cells migrate, crushed muscle extract is made. Crushing muscle releases factors in response to the damage. The crushed muscle is filtered, and the remaining liquid contains the important motility factors. This extract is then divided and flash frozen in liquid nitrogen.
We're now going to store our samples at minus 81 degrees Celsius. This helps the proteins from denaturing.
The next day, the cells are analyzed under the microscope to make sure they are alive and healthy. The cells are then moved to a new plate with fresh media to select for only healthy cells. The cells are then incubated for 2 more days. A new 48-well plate is coated with the satellite cell's native substrate. This substrate is required for the cell's movement. A sample of the cells is analyzed using a hemocytometer to estimate the number of cells you have. A small grid is present, and the cells within the grid are counted. At this point, the cells have proliferated enough to use for the experiment. 10,000 cells are then plated onto each well of the native substrate-coated plate. The stimulus factors, contained in the crushed muscle extract, are then added to the cells in each well. These factors are believed to make the cells move. The plate is then placed on the time-lapse microscope. This microscope captures the location of the cells every 5 minutes.
Now I'm going to pick positions for my cells. My cells are plated on a 48-well plate, so I can test different conditions in each well. What I want to do is find the cells, get them in focus. Once I have a good position, I will set it on the computer.
The images are compiled to create the video of the cell's movement. The time-lapse microscope uses different wavelengths to capture images of the fluorescent cells. This allows researchers to track the satellite cell's movements. A better understanding of satellite cell movement is important for both basic science and understanding degenerative muscle disorders.